This is a production of Cornell University.
All right, thank you, Anna. My name is Marjorie Hanneman, as Anna said, and I'm a second year in the GORE lab. And my research project that I'm focusing on today is the analysis of vitamin B3 or niacin in maize grain. To give a cultural context to vitamin B3 in maize, we need to talk about nixtamalization, which is a traditional cultural practice from Central America where the maize kernels are cooked in lime and then eventually ground to create a masa to make tortillas or tamales or other foreign-based foods. One thing about this process, though, is that it increases the bioavailability of vitamin B3. And in maize diets, where that's the predominant food, there are, in cultures that don't have nixtamalization, we see severe B3 deficiency, and diseases such as pellagra occur, and that is predominantly because vitamin B3 is mostly unavailable in the maize grain. I have this pathway here showing nicotinic acid or niacin, which is our main form of B3, and a trigonalene is the N-methyl derivative of niacin, where this compound is inactive and undigestible because of this methylation. And in the majority of corn kernels, this is predominantly the form of B3. Vitamin B3 is also essential, specifically to the NAD cycle, where living organisms get the majority of their energy. I have the three compounds in the previous slide listed here, showing that trigonalene is an offshoot of this, this cycle, more of a byproduct. And there's some thoughts that it's involved in plant growth hormones, but for our cycle we're focusing on here is vitamin B3. It is inactive and is an offshoot from the necessary cycle. So to further look into the B3 compounds in maize grain, we used high throughput LCMS to phenotype these three compounds that I had mentioned in the cycle. Looking at about 1,500 different maize lines, we can see that there were three specifically that have extremely low trigonalene, and that would be the blue, green, and red triangles, specifically T232, which we will focus on, is a breeding line out of Tennessee. Those same three have an elevated level of nicotinic acid or niacin, the active compound. And so that's suggesting that something is happening between the nicotinic acid and niacin step and trigonalene. Also interesting, though, is that for nicotinamide, they're all at average levels. So we're wanting to focus on specifically what is happening between those two compounds. And our main question for this project is what is genetically causing this extremely high niacin level and low trigonalene level in the T232 maize grain? Comparing the T232 line, B3 composition with B104, which is a fairly standard line that we chose because of its ability to be transformed, you can see the difference in trigonalene levels, where it's basically a sliver here in T232. So choosing this average line in an extreme phenotype, we crossed them and self to make an F2 population so that we can identify causal variants that could be responsible for these unique rare phenotypes. Within our F2 progeny, this is a distribution of the trigonalene phenotype. And it's very interesting because you can see that there's a 3-to-1 ratio here to presence absence of trigonalene, with a 0, absolutely no trigonalene, compared to where B104 is, where it is a continuous distribution. This would suggest that there's some sort of genetic switch that has a homozygous recessive allele that is causing an absence of trigonalene, and then to have a continuous quantitative phenotype for presence. Then genotyping our F2 population with RHAMP-seq and then inputting it into an ICIM software. We were doing a QTL analysis to look at the 3 phenotypes we were interested in. Now, we specifically wanted to look at trigonelline, where we had a very large QTL on chromosome 4, and also seeing it in nicotinic acid. So that would suggest that there is a single locus that's going to be associated with the niacin to trigonelline pathway. Now, nicotinamide also had a few QTL peaks, but we are specifically going to be interested in only nicotinic acid and trigonelline for now, and look into that later since it had more average phenotypes for our extreme lines. This QTL was also about 2 megabases wide with 51 genes in it. And then we are hoping to narrow that down and think and identify genes that are specifically involved in methylation. And specifically, here is our NAD cycle again. What is causing this to stop? So for our next steps with this project would be to integrate our QTL results with GWAS, TWAS, and EQTL, specifically from those 15,000 lines we originally looked at, to see if there are any genes that would be hit here. But because this is a rare phenotype, it would be interesting to see what else is going to be identified. Additionally, we'll use orthologous information from other model plant species, such as Arabidopsis, to prioritize our selection of possible causal genes. Then using de novo assembly of T232 genome, we can identify the causal variants and see if they are also present in the other 2 extreme lines. And I would like to acknowledge my committee members, and specifically the Gore Lab and our collaborators at Georgia, Michigan State, and Iowa State. Thank you.
Okay, I got a simple question. For the biparental population you develop or that you use, do you know why you use the average line instead of another extreme that had a higher level?
Sure. So the question was, why did we choose an average line to make the biparental population instead of using 2 extreme lines? That was because the average line we chose, B104, is transformable. And so then later down the line, we would be able to use that line to use gene editing or something else to confirm causal genes.
I guess I was gonna ask, what do you know about the background of the line, and do you have a hypothesis as to why this is so rare?
The background of the line was it was a traditional breeding line from Tennessee. It was a non-stiff stock line. All 3 of the extreme lines were not the same heterotic group, though. I am thinking that this is a mutation in a single gene, and that could be resulting in that it continued. There's not seeing it in any other lines. Could be because there wasn't enough of a mutation to have a selection over time.
So Kelly, just to add, the mutation doesn't appear to be lethal, but it's just occurring at very rare variant frequency.
So maybe like a more recent mutation, or there's just no selection.
I would say, well, probably a combination of both. Yeah. Or if it did happen, say 80,000 years ago through drift, it could have been lost, but it probably occasionally occurs. Yeah.
You had your hand first.
Oh, so you said it's not a lethal phenotype. Does it show any other phenotypes, or just the plant itself, other than the different levels, accumulation levels? Does it look pretty much the same?
The question was if the plant otherwise looks the same because of the extremely low trigonelline lines. Specifically T232, there's not anything obvious. I want to look more into how does if trigonelline is going to be used as a plant hormone, how does that affect growth? One of the extreme lines, G14, though, is very difficult to get seed from.
Oh, I'll just add that this is a seed phenotype, but we haven't looked at in other tissues outside of seed. So that's an unknown. Yeah.
Is the trigonelline an anti-nutritional factor? I mean, is it bad to eat? Outside of the fact that it prevents you from getting vitamin B3, I guess, but.
Not that I know of, it's just undigestible and unprocessable. It goes through a human body. I don't know of any negative. Well, thank you.
This has been a production of Cornell University, on the web at cornell.edu.